Good morning, everybody. I'd like to welcome everyone to the panel discussion this morning on translational health research. While we'll just give another minute or two for those who are fashionably late to join the presentation, I would like to ask the panelists to turn on their cameras so that the audience can see who we are. Thank you, everybody. So I want to give a little bit of context to what this morning's presentation is going to be about. And the focus is going to be on translational health research and how it plays a pivotal role in uniting basic science, veterinary medical research, and human health research. And as a part of that, companion animals, particularly cats and dogs, they share many diseases and disorders with humans, ranging from cancer, cardiac disease, psychiatric disease, orthopedic disease. And our pet animals also have tremendous genetic diversity, which is frequently lacking in our preclinical models of disease. And also, they share a complex environment with humans. So they're an almost perfect model and also a largely untapped opportunity for the development of new disease treatments. And as a component of One Health, translational health research spans multiple disciplines. And in this panel, you're going to hear from a variety of researchers who work in different scientific disciplines and how they bring their discoveries out of the lab and into the clinic to develop new treatments for the benefit of both animal and human health research. And it's an often underestimated benefit from this translational research where the primary focus is humans, but in reality, we get all of these new treatments for our pet animals. So with that introduction, what we're going to do is we'll start with introductions followed by some rounds of discussions. And I'm going to try and keep everybody on track in terms of time. For the audience, if you wish to ask a question, please post it in the chat channel. And we've got some time allocated at the end for questions. And we'll take questions from the chat channel. Also, if you've got a burning question or comment to make during the discussion at the end of the session, then just raise your hand using Teams, which I'm sure everybody knows how to do. So we're going to start by introductions, and I'm going to introduce myself first. I'm Howard Dobson. I'm a veterinary radiologist. I'm Professor Emeritus in the Department of Clinical Studies. I now work as an independent consultant in research. During my career at the OVC, a lot of my research was translational in nature. Now, as a consultant, where primarily I work for a large global imaging-only contract research organization called InVicro, where we do imaging-based research ranging from cell culture through small animal models, large animal models, and then translation into people, where we go right through to late-stage clinical trials. So everything I do in my day-to-day work is focused to the ultimate translation into the clinic, even though the work that I do as Senior Director, Discovery and Preclinical Sciences, what I do may involve rodents, or specifically for me, non-human primates, other large animals, including dogs, pigs, and sheep to some degree. So everything is focused towards translational to the clinic, and certainly in the industrial context where I work, working with most of the large pharma companies and many startups, a huge amount of this work, the hot topics at the moment are neurological disease and cancer. And cancer in particular is a real big one with some exciting developments that are coming from institutions like the University of Guelph, where a lot of benchtop research is being done to further human health research. So now what I'm going to do is ask each of the panelists to introduce themselves. I'm going to start off by introducing them by their formal title, but then from there on, I think we're going to work on first name terms only, because this is an informal discussion. So no stuffed shirts today, even though I might be wearing a tie myself. So I'm going to start with Dr. Tony Mutzis. So Tony, if you'd like to go ahead and introduce yourself, please.
Sure, Howard, I will do that. My name is Dr. Tony Mutzis. Hopefully everybody can hear me okay. I'm an associate professor here at OVC and a veterinary medical oncologist by training. I have an appointment in clinical studies as well as the Department of Biomedical Sciences. And I guess for more complete introduction, how did I get where I am? I did my DVM here at OVC, so I'm an OVC alumnus, and I went into private practice for a year, mixed animal practice, as I told myself I would, in Newcastle, Ontario, and had a great time, but already recognized that I was a medicine mind and I wanted to go back and specialize in a medical discipline. Was able to come back to OVC and did an internship here, and then did a residency in medical oncology at Purdue University. After that residency, I was able to get a clinical instructorship at the University of Queensland in Australia at the veterinary school there, and then came back to Canada, started a PhD at the University of Toronto at Sunnybrook Research Institute through their Department of Medical Biophysics in Bob Kerbel's lab. And then at that point, I was lucky to be able to start consulting for OVC one day a week through their developing oncology service. I did a brief postdoctoral fellowship after the PhD concluded at the University of Melbourne, the St. Vincent's Institute there, and at that time was able to arrange the current faculty position that I've been in now for about 10 years.
Thank you, Tony. Our next panelist is Dr. Jim Petrick.
Thanks, Howard. Yeah, good morning, everyone. My name is Jim Petrick. I'm a professor in biomedical sciences here at the University of Guelph and lead a translational cancer research program where we're focusing on the discovery of novel therapeutics for the treatment of advanced stage cancer. So we've focused predominantly on ovarian cancer historically. Now we've branched out. We're looking at our therapeutic approach in pancreatic cancer as well. And we're really excited about the work we're doing. Those are both very devastating disease, and particularly devastating because they're not usually identified or detected until advanced stage. So lots of really smart people are working at early markers of disease and early detection, but the clinical reality is that most people aren't diagnosed until they're really sick. So we're looking at it from the perspective of the therapeutic opportunities to help people in the advanced stage when they already have fairly aggressive disease. So we're really excited about some of that work. I'm also the co-director of a Bench to Bedside Translational Research Institute, where we're enrolling, where one of the important components of our research plan is to bring in companion animal cancer patients in the therapy development pipeline. And I think there's some really exciting opportunities there to enhance the efficacy and the success of some of the cancer therapies that people all across Canada are working on. So really excited for this morning. I think it's a great opportunity. I appreciate the opportunity to be part of it. Thanks, Howard.
Thank you, Jim. Next is Dr. Christopher Pinard. Christopher, take it away.
Thanks a lot, Howard, and hopefully you guys can hear me okay. I'm Dr. Christopher Pinard. I'm a medical oncologist here in Toronto, but also hold a postdoctoral fellowship position at Sunnybrook Hospital. I actually trained under Dr. Mutzis just over a year ago, so just finished recently. He wasn't too hard on me, but overall I did my OVC training or DVM training at OVC, did an internship in downtown Toronto, and then did a clinical trials internship actually at Colorado State University, and then followed with my residency at Guelph, and that has led me to where I am today. So really trying to work both between Sunnybrook, OVC, and in the current practice that I'm at, trying to bridge academics and private practice, and really bridging kind of the human and animal gap with a lot of cool technologies, artificial intelligence, and genomics as well for cancer treatment in pets.
Thank you, Christopher. And next is Anne-Steffi Ram. So Steffi, would you like to introduce yourself?
Hi everyone, I'm Steffi. I'm a PhD student in the Department of Biomedical Sciences. My research interests are having to do with surgical oncology and near-infrared imaging. So I'm currently completing my thesis with Dr. Petrick and Dr. Oblock, which focuses on developing a cancer-specific imaging agent to identify tumors and metastatic lymph nodes during lung cancer surgeries. And yeah, it's pretty short, but I don't have a lot of qualifications yet.
but you will. You're in a great program.
But I will.
So what I want to do now is start into our panel discussions. And the first topic that I want to introduce is understanding translational health and clinical research. So Jim, could you help those unfamiliar to translational health research as a concept understand what it is?
Yeah, absolutely happy to, Howard. So the way I think of translational health research is utilizing a variety of models. My lab historically has been very interested in modeling disease, and I think that that's one thing we have to pay particular attention to is the quality of our models. And so translational health research to me is using the appropriate models from varying different species to help answer complex biological, biomedical questions. So in my lab, we historically have looked at rodents as models of disease, and we've done a lot of really, I think, innovative work in modeling cancers in particular in our case. But we also realized that there's a lot of shortfalls. Every model has shortfalls, and we've identified there's a real opportunity here at Guelph in particular with involving other more complex models that have spontaneous disease, such as our canine and feline companion animal cancer patients. So we translate our findings from our preclinical rodent models to a more complex, more advanced biological model, such as our cancer patients. And then ultimately what our goal is to translate those findings into the human clinical space. So I envision translational health research as a way to take a biological question, identify it with your basic science techniques and identify mechanisms, and then take it through a translational paradigm where we look at relevant models in a translational ontological perspective, where we go in more and more complex models, ultimately to our eventual clinical goal. So an important part to me of translational research is ultimately it has a clinical application. Otherwise, we're looking at basic science, which has a lot of merit on its own. But translational research, really, to me, I think, has a clinical trajectory. And what your ultimate clinical goal is will really dictate what your translational models might be. So I think, some of the important things to think about are, what are the biological questions you're trying to address? What are the appropriate models? We all fall into the trap of, we have a model, so let's see what we can do with it. And that's really counterintuitive to me as a scientist. I think we need to have a really strong, solid scientific question and then identify the appropriate models to help answer that question. So, just to recap, translational health research to me is identifying a biological question that has a clinical trajectory and ultimately will address a clinical need, and then bringing in the appropriate resources and animal models, et cetera, to help bridge that gap from basic science idea and mechanism to clinical application. And what do we need to do translationally to fill that gap and answer the questions in a really robust way that ultimately leads to answering that clinical question that we have.
Great, thank you, Jim. Tony, could you help us with the definition of clinical research and comment how it fits within translational research?
Yes, sure. Thinking about the definition broadly, I would say, clinical research involves naturally occurring disease. And it's interesting to say it in that form because we use that title, naturally occurring disease, oftentimes when discussing our research in companion animals with diseases as models, similar to what Jim was saying, and how they may be appropriate for application ultimately to human clinical research. But in the context of veterinary medical practice, those are clinical research approaches. So there's clinical research, whether that be observational or interventional or any of those categories. If it's happening in the naturally occurring disease setting, cancer or otherwise, then in my mind, even prevention research and all these aspects are done as an umbrella of clinical research. And then within that, if you focus on the interventional aspect, whether they're drugs or other things, drugs being my sort of bias, then you're into the sort of clinical trial aspects of clinical research. But as an overarching term, I think if the disease is not a model in and of itself that occurred naturally in whatever species, and you're taking samples or doing interventions or observing that biology, then you're performing clinical research. And then it's intertwined with the translational aspects, as Jim was saying, when we're trying to apply those that knowledge that's been gained in the basic setting and in the preclinical modeling setting, however good those models may be, into that setting of naturally occurring disease, then there's that intertwining of translational research with the clinical environment. So that's how I would broadly define it.
Great, thank you. And if I can just make a comment here, I think traditionally in veterinary medicine, we've looked at what's being done in human medicine and then back translated it to our clinical practice. Whereas now I think we've switched that around and we are leading the charge and developing new treatments in veterinary medicine, and then they're being translated forward to human clinical practice, which I think is a wonderful development. So what I'd like to do now is I'd like to go around the panel and ask each of you to provide an example of how your research or practice fits with translational and clinical research. And I'm going to start with you, Jim, if that's okay.
Yeah, absolutely, Howard. So yeah, as I said before, I kind of promoted myself as a translational researcher. So what does that mean? I think, as I mentioned a few minutes ago, we've gained a lot of traction and momentum in our basic science using genetically competent, immunocompetent mouse models of the cancers that I mentioned before, ovarian cancer and pancreatic cancer, and we've addressed and answered a lot of really important, I think, innovative biological questions, and shown that our therapeutic approach, at least in the preclinical models, has some real interesting findings, and we have preclinical impact, and we're excited to take it to the next step. So what the next step for us, and I'll kind of lay out what we're doing as a bit of a roadmap of how we're addressing a translational approach to cancer therapy development. So we've identified a molecule that does really interesting things and enhances the uptake and efficacy of other therapies like chemotherapies, oncolytic viruses, immunotherapies, and we've identified that in our rodent models that there's a lot of impact there, and we can have curative outcomes in advanced stage ovarian and pancreatic cancers, which is exciting. The next step for us is we want to now enter into the clinical trials, companion animal clinical trial space. And so the first step that we've done is we've done a healthy dog study to look for safety. So we've done a safety study in healthy research dogs, and the safety profile was very good. And now we're hoping in the very near future to embark on companion animal clinical trials, looking at a couple different sarcoma models, so hemangiosarcoma, soft tissue sarcoma, and then eventually osteosarcoma, as proof of principle and to show clinical efficacy in the companion animal models, which was, once again, to reiterate, these animals show up with spontaneously occurring disease, that they're not contrived models like we've had to do with our rodents. So they show up with disease, they live in the same environment as us, they're under the same environmental pressures. So and a lot of data shows that their tumors and their disease profiles very closely mimic what we see in people. So our next step is to go to companion animal clinical trials, and then ultimately from that we have partnerships with Ontario Institute of Cancer Research and their Triphase program, where we're hoping to fund phase 1-2 clinical trials in women with advanced stage ovarian cancer initially, and then in men and women with advanced stage pancreatic cancer. So that's our trajectory, and we're really excited about that. So that's how we're addressing this translational approach. And the one thing, just to jump on the soapbox for a minute about the importance of including these companion animals in our therapy development pipeline, is that right now in cancer, when we take our current way we develop therapies is to take an idea, in vitro modeling, we take it to rodent models, and we have an outcome like we've had with our ovarian cancer and pancreatic cancer. And then the current model now is to translate that to phase 1 trials in people. So we jump this translational gap where we go from rodents right to humans. And necessarily that's the way it's done right now, but the reality of it is, in cancer specifically, that results in a 94% failure rate of therapy. So 94% of these therapies that we're developing in this current therapy development pipeline fail to have any clinical benefit whatsoever, which has in my mind, two really main consequences. One is an economic consequence. We're spending millions and millions of dollars to get to a phase 1 clinical trial, and 9 out of 10 of those therapies are going to fail. So the health, our health research funding is not being spent in a really economically viable manner. But more importantly to me is that we have sick people that aren't getting better. We have people with cancer that we're not developing effective therapies. And here, our mindset is, let's bring in these companion animal cancer patients, develop therapies that work in their cancers. That'll allow us to refine those therapies and take really robustly vetted therapies to human clinical trials. So increase the chance that we're going to have a real successful therapy. But also importantly, we're at a veterinary college. We're developing these therapies and we're testing them in our companion animal patients. But the other benefit is that we're going to develop amazing cancer therapies for pets. So we have this bimodal impact in effect that I think will be really meaningful, because we're going to develop really robustly vetted, excellent curative therapies that are going to benefit both our pets and people in the clinical trial space.
Great. Thank you, Jim. Christopher, what would you like to add to this discussion?
I think Jim really kind of summarized a lot of the benefits of translational research and kind of how that fits under the One Health umbrella. And to kind of reiterate again, we see a very naturally occurring model, and as Jim said, that definition seems a little bit arbitrary, and I think Tony touched on that as well. But these are not models that we are inducing cancer in, or mice that we're injecting cells into. And I think when you talk to a lot of human clinical researchers, they're thinking that translational oncology is really let's look at or answer a research question very simply in the lab by looking at it in mice and trying to then immediately translate that to people. And that probably leads to a lot of the complications that we see with getting drugs through the pipeline. And so I think, again, to Jim's point, something like comparative oncology or translational health research in oncology really could accelerate those things. And to just extend on that point, I thought I'd give one example of exactly where that has occurred. And it was a project that we were working on when I was at Colorado State University during my internship, in that there was, at the time, a big problem that dogs with osteosarcoma that had metastatic disease really had no great treatment options. And we had tried therapies, something called Palladia, that in our world is a common anti-cancer drug that we use, and there's similar drugs that they use in people, but on its own didn't really seem to work well. And so basic science researchers went to veterinarians that, they knew they had this oncology, comparative oncology program. They were able to repurpose a blood pressure drug, of all things, that essentially altered the immune response in the tumors. And we actually saw distinct and really amazing responses to patients that had metastatic disease. And that's actually been so profound that there are children in Colorado now with metastatic bone cancer that are undergoing clinical trials with this exact same therapy. So I think that really just speaks to the utility of, the opportunities that we have in a translational health program.
Great. Thank you, Christopher. Steffi, what are your thoughts on this point? You're on mute, Steffi.
Sorry about that.
No
So
problem.
it summed up really well that how animals have been used to understand human disease and then vice versa. So an example of that in my thesis is that we've been using like canine cells and human cells and verifying what overaccept receptors for lung cancers are present in both. So that comparative approach is going to be used to use our imaging agent in both canines and humans since it's a targeted agent. So using that first basic science in cells and then translating it to companion animals before human trials really helps us understand how well this works and, you know, identify any pipeline barriers that will end up with any targeted type of therapeutic. So, and it's really great because lung cancer tumors, they resemble human non-small small cell lung cancer histotypes, and they're naturally occurring, and they occur just the way as humans who are never smokers get lung cancer. So I think it's a really great model, and using that and, you know, utilizing that opportunity is going to push targeted therapeutics even further.
Great, thank you, Steffi. Tony, a few final words on this topic before we move on to the next one, please.
Sure, yeah. Happy to provide another example. I can think from my residency training way back where my advisor, Dr. Deb Knapp, found an anti-cancer effect by palliating dogs that had end-stage cancer, similar to what Chris described, with an anti-inflammatory drug, piroxicam, that is used as a musculoskeletal drug. People may be familiar with that whole category of agents. And she saw one case of a dramatic response and then kind of pursued that to the point where she was able to evaluate it in a lot of different veterinary cancers, found that bladder cancers in particular had a good response to this drug, and at the time was discussing with the establishment about whether this was an anti-inflammatory effect or indeed a true anti-cancer effect. And by the time I started my training, we were doing scopings of all of our bladder cancer cases and became a satellite for a lot of bladder cancer case referrals from all around the Midwest, where we would take a sample, treat for 4 to 6 weeks with the drug, take another sample on treatment, and then analyze what was happening and able to show direct anti-cancer effects and anti-angiogenic effects, and really get a better answer as to what was happening at this level. And it was helped by the fact that at that time, there were some studies coming out about people predisposed to colorectal cancer and aspirin therapy and polyp reductions and things like that. The research got to the point where the Indiana University School of Medicine was very interested in seeing, at that time again, Celebrex and Vioxx were these brand new drugs that everybody thought were really good cyclooxygenase inhibitors. They had a series of patients who would have superficial bladder cancer that would relapse, and they were destined to have complete bladder removal, cystectomy. And there was a window of time of about 4 to 6 weeks between the time they were progressive and they were going to have that surgery. And so the research at Purdue led to them integrating a COX inhibitor trial in those patients before they had their bladders removed, so that they could ultimately as well look at what the biological effects were on those bladder cancer samples. And so as a model of bladder cancer for humans, and when you're interacting with the human clinicians in that way, it really drives home the One Health, One Medicine aspect of the work you're doing. And so that for me back then was sort of a motivator for how I would go on for doing research and grad training from there.
Great, thank you, Tony. Jim, what kind of diseases and disorders do humans and animals share that could be nicely addressed through translational health research?
Yeah, well, that's, to be honest, Howard, that's $1 million question, right? I kind of expounded the importance of making sure that your model is a robust model, and I think that's where we are right now is really trying to identify what are some of the diseases that companion animal patients and human patients have a lot of fidelity between. So, we look at cancer, and, cancer is a big term. So even refine it further, we look at specific types of cancers. And for example, I know Tony and Jeff Woods and Alicia Velotti-Petit have been doing a lot of work on osteosarcoma with the dog bone project. And we know that there's a tremendous amount of, genomic fidelity, morphological feature similarities, et cetera. And we think that the dog osteosarcoma model is actually an excellent model for studying the disease in humans. Similarly, feline mammary cancer. feline mammary cancer actually models triple negative breast cancer in women very, very well, which is triple negative breast cancer is a very difficult disease. The one benefit that we see in dogs, so we've had the criticism, given Tessa we've been working in this space and writing some grants that, that companion animals get a lot more sarcomas, whereas humans get carcinomas. So, are you really modeling human disease? And in some cases, so feline mammary cancer is a carcinoma that replicates human triple negative breast cancer very well. But one of the real strengths to that argument, against this translational aspect of this approach is that sarcomas in humans are incredibly rare. So some people say, you're using models that may not replicate some of the most common cancers in people, and that might be true. But looking at some of the sarcomas in people, because they're so rare, they're incredibly hard to study. There's very little known about the biology, response to therapy, etiology of some sarcomas in humans, where we have animals that show up with tons of sarcomas. So, and we know that there's a fair bit of fidelity between those diseases. So we view this as an amazing opportunity to robustly increase our knowledge base about the biology of disease and sarcomas, potential response to therapies, in a way that's impossible to do in humans because of the rarity of the diseases. So, we think that there's a lot of translational power behind utilizing our companion animals to look at rare disease in humans. And, I'm coming from a cancer-centric space because that's what I work in. But we know that there's a lot of cardiovascular disease, a lot of neuropathies, for example, that are shared by our companion animals. So, as we're working on our translational work in the Institute under the cancer umbrella initially, our goal is to branch out and bring in all of our collaborative research areas, so cardiovascular disease, neurological studies, regenerative medicine, under the umbrella of the Institute to help facilitate this translational clinical trajectory of the research. But I think that you asked a really important question: is what is the ultimate utility of the animals? Once again, studying the animals for the ultimate sake of improving animal health is a very righteous and important goal on its own. But if we want to further the translational opportunities and look at the impact on human disease, I think that we have to spend the time. We can't just assume that because they have spontaneous disease and they're more complex, genetically heterogeneous animals, that they're going to be better models for human cancer. We have to do our due diligence, and we have to look at how similar are the diseases, and if they're not similar, then they're not a good model, and we can't really robustly include them in the therapy development pipeline. But I think in my experience, in what we've looked at so far, there's incredible opportunities for us to learn more about the disease and really improve and enhance our therapy development as we move across that whole translational spectrum, ultimately to human clinical application.
That's great. And just a comment from my perch in industry working closely with pharma. What I always find surprising is there's a huge amount of interest in rare diseases. And I think the example you quoted of the sarcoma carcinoma issue is a perfect opportunity for the model the common model in animals of the rare disease in humans. And I think there's an exciting opportunity there. And then just as a question, Jim, given that currently there's a lot of thrust on the genetic background of diseases, and particularly cancer, what do you think the merit is of doing, large-scale investigations of the genetic background of a range of diseases in humans and animals, and identifying which animal diseases are going to be good models of human disease based on the commonality of the genetic background?
Yeah, that's a fantastic observation and question, Howard. I think that's not only is there merit to it, I think it's utterly critical that we do that. We need to do that high level, broad spectrum omics approach to look at the strength of predictive models. So one of the things that we have at the University of Guelph and OVC is this companion animal tumor sample bank. The CATSB it's actually the only repository of companion animal cancers in a tumor bank database in the country. In partnering with Ontario Institute of Cancer Research, they've become so excited about this opportunity that they're investing in developing a dashboard. So it's a searchable dashboard that links our companion animal tumor sample bank with their Ontario tumor bank that OICR has developed, which has tens of thousands of human tumor samples. And what their goal is to create a searchable database or dashboard that links the two tumor banks. And then we can start asking those questions in a really robust way. As we can look at canine osteosarcoma, for example. We have all these samples. We have liquid biopsies. We have tumor samples. We can start asking questions, from the companion animal space, and then applying it to what they've figured out, or what they've been able to identify with the human tumor samples. And that's an incredible opportunity that's unprecedented. Nowhere else do we have a really defined link between companion animal resources and human resources, and we can start asking those questions that you alluded to with your question, Howard. But then beyond that, I think one of the goals that we have with the Institute is to really expand that opportunity. And in the veterinary space, we haven't been as aggressive in looking at some of the really broad spectrum RNA sequencing. In some of the human databases, as soon as they get a tumor, they'll do RNA sequencing on it, and they're starting to amass really huge amounts of data and data sets. I think we have the opportunity to do that here with the cost of some of those technologies coming down to a more reasonable space. I think that we can start to really build large amounts of RNA protein sequencing data that will be very easy for bioinformaticians to start asking those questions about disease similarity, differences, predicted response to therapy. Personalized medicine is such a huge thing right now. So there's a lot of opportunities that I think we're really on the doorstep of getting towards.
Great. Thank you, Jim. What I'm going to do now is go around the panel and ask you all why you're interested in translational health research and what inspired you to go down this path. And I'm going to start with Steffi because you're the newest to translational medicine.
So basically during undergrad, I really liked the idea of collecting data and ideas from basic literature and then creatively applying that to disease solutions. So it's a really interesting way of problem solving. And so during my postgrad studies with Dr. Petrick and Dr. Oblak, they really facilitated this, where I needed to be able to get out of my comfort zone and reading literature and learning things outside of my field. For example, things like surgical oncology and near-infrared imaging, things I've never learned when I was an undergrad student. So things like that really pushed me forward to learn about translational medicine and how to incorporate different fields and becoming more of an interdisciplinarian in the way that you problem solve for diseases.
Great. Thank you, Steffi. Christopher, how did you get here?
Tony's probably heard this story before, but really it started back in vet school for me when Dr. Paul Woods, one of the other medical oncologists at Guelph, who a lot of you probably know, brought in an owner with their dog who had recently been treated and diagnosed with lymphoma, and they were talking about the translational relevance in that a lot of lymphomas in dogs looks very similar to non-Hodgkin's lymphoma in people, and not only were they studying it in this particular patient, but this patient did extremely well. And so seeing the benefits there, and then also personally in my own personal life being impacted, not me with cancer, but family members and so on throughout my training and so on, you really start to see where there's a huge benefit in partaking in something bigger than yourself and bigger than a small benchside project. But that benchside project could really amalgamate into something that could really change the lives of other people and pets, and that's a really unique and cool opportunity.
Thank you, Christopher. Tony, what was your route here?
Oh yeah, my experience really does mirror what Chris said. I think going back to being able to specialize in a clinical specialty, I did that again based on my interest and aptitude for medicine and medical oncology, and then you very quickly get the appreciation for some of the larger investigational research questions that can come out of that. And even interviewing for residencies, I remember I interviewed at Wisconsin and got to sit in an office with Greg McEwen, who's one of the fathers of oncology, interviewing you for a position, and it's just like, you know, talking about some of the things he had been doing and already at that time. And then, you know, that was probably the best interview of my life. I loved it, but I didn't match at Wisconsin, so maybe it wasn't as good for Greg. I don't know. But the result at Purdue also was really a springboard for me, because compared to OVC and other schools, the caseload there was comparatively quite low. So if you were there and wanted to see as much oncology as you could to get out into a practice in New York City or somewhere, you know, we saw everything, but we saw a lot more bladder cancer because there was a very dedicated research interest in that area. And for someone like me, that was a big inspiration to continue on, as I sort of mentioned with the experiences before. So by the time I was finishing the residency, then that inspiration was there to continue on for more training.
Great, thank you, Tony. Jim, what was your journey?
Yeah, my journey actually has been a little bit varied. You know, I came to Guelph to do a postdoctoral fellowship with John Lamar to look at angiogenesis, so blood vessel formation in the ovary from a reproductive perspective. And I was really interested in looking at the factors that regulate ovarian function in women, obviously, and you know, factors that could control that. And I was really interested in looking at it in the bovine model. I've always been really, hopefully, attuned to the relevance of good modeling, and I really wanted to look at the bovine model as a translational model for human reproduction. And I'll tell you what, Howard, the granting agencies were not having any of it. When I presented, you know, the research approach and experimental plan using bovine models, they came back and said, "What are you doing? Like, why aren't you looking at this in a mouse?" And I said, "Well, because I'm not an idiot. Like, the mouse is the most horrific model for ovarian reproduction to translate to humans, because they're nothing similar. Whereas the bovine model actually is an amazing predictive model." But I'll tell you what, I never was able to overcome that resistance. We did a lot of great work, and we found a lot of really interesting things, and you know, that angiogenesis approach, you know, serendipitously translated to my interest in ovarian cancer, based on some interesting findings back in the day. But you know, that always left me with a bad taste in my mouth about not being forced into using models that were not relevant. And you know, I was really thought I was being innovative and proactive, and we've done a lot of amazing work with rodent models, and they definitely have a place. But you know, now as we move on to other models and involve other models in this translational spectrum, I'm really enthused and excited about those opportunities. We'll always use our mouse models, and we'll always learn really important biological mechanistic things. But you know, I've always wanted to go back to those reviewers and say, you know, like, because there has been, you know, Dr. Wiltbank down in Michigan, and you know, they found some really amazing predictive, translational, reproductively impactful things using the bovine models. And you know, I'd like to go back and say, you guys need to open your minds and be a little bit more attentive. And you know, you know, I think we'll have an opportunity to talk about, you know, advice to grad students and things like that. So I'll save some of those comments for down the road. But I think that it's not a trivial thing to really pay attention to what you're doing and the models that you're using. So my journey now to this translational space is, I think it, you know, really galvanized my my realization of the importance of being really mindful of the work that you're doing. And we can fall into the trap very easily that, you know, our lab does this and we use these models, and lots of times those are great, but you know, we need to make sure that we're using the appropriate tools to answer the questions that we have in front of us, as opposed to looking for questions that we can use our tools for.
Yeah, great, thank you, Jim. And what I hear from all of this is that, you know, when you're early in your career, you've got to kind of follow the established path, because the off-the-wall idea is going to get the same response that Jim get. What on earth are you talking about? Whereas when, you know, the likes of, you know, Jim and Tony, who are more established in their careers and have got a track record, then you can start coming out with the you know, the innovative ideas. But I think it's very sad that you know, the innovation from young people who at that stage are actually very innovative, and sometimes it gets repressed because of our conventional granting process. And I'd like to think that in industry there's a bit more of an open mind, but I'm not sure that that's necessarily always true. So with that, I want to take us through to a slightly different approach to this. And Tony, could you walk us through the therapy development process, please?
Sure, yeah, I can give a broad overview for the audience, really, and then we can talk maybe specific components or whatever relevant. But I think of drug development first off with therapy discovery. So there's drug discovery and then development, and the research that goes into discovery is wide open, right? That can take so many forms. That's the basic science Jim talked about and everyone else, where the fundamental discovery is made that someone then brilliantly comes to the conclusion, maybe this has an application, a therapeutic application. Where do we go from here? And that's when the development process begins. And depending on the nature of the research group that's thinking about this, there are regulatory considerations that come into play at multiple stages, including Health Canada and the Veterinary Drug Directorate and FDA, USDA, depending on where you're at. And I'll sort of leave the specifics of those aside. But when it comes to the drug development, I'm going to again use the word drug because that's where my space is. But it's one thing to have a target or an interesting lesion you're optimistic about, but there's a whole other area of research into converting that into a therapeutic. And the medicinal chemists or the biologic people making antibody specifics or whatever form your drug's going to take, there's a whole body of research going from that discovery point to making it into something that people think can be administered to animals, even in the preclinical sense, let alone down the road in the clinical evaluation steps. And so, I remember in medical biophysics, there were three components. There were the medical physicists doing a lot of the imaging research. There were the cell and molecular biologists group. And the third wing, which was the smallest, were the structural biologists who didn't have as many grad students. But when they were talking about all of those drug libraries and protein pockets based on charge and all these other aspects that they were interrogating in order to come up with a drug that then goes through the process of evaluation for target modulation and, of course, toxicity and dose finding studies, all in preclinical models that, again, are driven by an investigator's interest in where they think this thing may be applied best. And then there's that that valley Jim talked about, the valley of death, where you've got a lot of preclinical evidence and you have some big decision making to do. When that jump is made to the phases of clinical trial, be they in human, skipping perhaps some of these other relevant aspects we're talking about, that's a dose finding study and a tolerability toxicity study in multiple, again, in my space, cancer patients of multiple types usually have gone through standard of care and are getting this phase 1 application to see whether we can hit on a optimal or even maximally tolerated dose. And then again, in the cancer space, phase 2 trials are then in the place that you think that therapeutic is going to be most valuable. Bladder cancer, for example, or a specific indication, and that may be where your regulatory claim is made if those trials are positive based on finally a therapeutic benefit, usually a tumor change in volume. And then ultimately the phase 3 clinical trial is that one that has the standard of care going up head to head with the interventional or investigational provisionally approved drug. And then I guess I'd end on a very important phase 4 aspect. So even after drugs are approved and we have new therapeutics on the market, there is a lot of post-approval monitoring in what we call phase 4 to make sure that even in the phase 3 trials, which go from cohorts of 3 patients at a time in phase 1 to approximately 24 patients in phase 2 to hundreds or 1,000 or more in phase 3, we're now in populations of people in phase 4 looking for other things that the drug may be associated with that could be problematic. And so, the nature of therapeutics takes many forms, from chemicals to antibodies to other biologics, but the process is somewhat similar along the way from my perspective. So hopefully that sort of lays a bit of groundwork.
Great, thank you, Tony. So what I'm going to do now is go around the panel and ask each of you, about the current challenges related to therapy development and how translational research will help. And maybe I can start with you, Christopher.
Sure, yeah, and I think, Jim and Tony have both hit on some really important points surrounding some of those challenges, in that, really going through the initial drug discovery, which is an exciting time, and preclinical phase is really great. And we can have upwards of, I don't know, for lack of a better number, 5,000 to 10,000 potential compounds that may work on X, Y, and Z, or whatever disease, receptor, whatever have you. But really only 5 of those treatments may actually reach the clinical trial stage. And then after that, only 1 may actually enter into a space where you can start to use that and put that in clinicians' hands down the road for patients. And the other problem is that the other challenge is that it takes forever to do that, especially in the human space. And when we're talking about diseases and treatments that work in the human space, people live or can live a long time with cancer nowadays. So it takes a long time to make those discoveries about whether we're actually seeing therapeutic benefit. I think that's been a huge issue from the immunotherapy standpoint, that we talk about these revolutionary treatments, but we're only gaining 3 months of disease-free survival by doing so. And so is that really in the benefit of the patient, and so on? But I'm digressing a little bit, but by doing these studies in the way that we currently use the drug pipeline, it can be up to 2 decades before we figure out one therapeutic that can actually be used in patients, and the millions or even billions of dollars that go along with that. So translational research then allows us to use our study of animals and good comparative models, and as Jim said, especially in a rare disease scenario, really accelerate that drug pipeline. And I think we have to be careful, because these are still animal patients, they're still pets, they're parts of the family, but at the end of the day, if we can benefit a human life by doing a clinical trial in pets, we can hopefully also benefit a pet life and future pets as well, and especially from a cancer setting.
Thank you, Christopher. Steffi, what are your thoughts on this topic? You're on mute, Steffi.
I'm so sorry again. So in terms of the imaging agents that we work with, a lot of the pipeline barriers that occur when we're trying to translate this to human trials is that there's a lack of standards, and it's not really helpful to model after what happens in a mouse model to a human. So really using companion animals is really helpful to kind of translate that, but also using companion animals to understand what are objective endpoint measures that we verified preclinically is really helpful, because applying those basic science principles into naturally occurring models, as we say, is very helpful to see what is similar to humans. Like, for example, we recently had a clinical trial with a nanoparticle called morphosomes, and what the objective was to learn how to model our clinical trials based on how the companion animal clinical trials occurred. So using information from one clinical trial to another to benefit another is, I think, really great on both parts for pets and humans.
Great. And I totally agree with you there, Steffi, because in my world, where everything I do is based on imaging and translation, if you're trying to do imaging work in small animals, in rodents, and then translate it to humans, just the size difference, quite aside from any physiological differences, have a huge impact. So a lot of the work is definitely aimed at large animals. Yeah. Thank you, Steffi. Jim, do you want to round out this little topic?
Yeah, absolutely. Thanks, Howard. So, from a basic scientist perspective, one of the major impediments to bringing therapies forward and going through the therapy development program is really a disconnect between basic science and clinical application in terms of what are the steps. As a basic scientist, when we started developing this compound and this approach, and we started looking at going into the clinical trial space, I tell there's a real lack of information out there for basic scientists to get to that next step. And what are the regulatory processes? What are some of the things you're going to need to think about, from a clinical trials perspective, from safety, efficacy? What are the the regulatory processes by Vet Drug Directorate, and Health Canada, like Tony mentioned, and they're very disparate. We had a meeting with Veterinary Drug Directorate yesterday as part of the institute work that we're doing. And they said these are the some of the things you need to think about when bringing a veterinary drug forward, or veterinary therapy forward, but they're very different from bringing something forward for human applications, and there's such a disconnect. There's such a disconnect between basic science and clinical science and clinical application, and then when you're looking at a veterinary application and a human application, there's still a chasm there as well. There's not a lot of crosstalk. There's we very naively went to them and said, is there kind of a roadmap out there for someone like me that can bring a therapy forward, and kind of a stepwise flow chart or tick the box or some type of program out there to help facilitate therapy development for someone that's not in that industrial space or that clinical trials. And it's a big challenge. I'm not going to lie. So, you and I have talked a little bit about it before, Howard, and I'm sure some of my questions must be comically naive for someone like you that have so much expertise. But we're at the space where we almost need to bring in consultants like yourself, Howard, to help us navigate the process, because it's a very murky road, and there's really no really good roadmap for doing that. So I see that as something that I think there's a lot of opportunity to really refine that and work on that, and make it a little less challenging to bring therapies forward and understand what we need to do. Because in the work that I'm doing, I've already realized some things that I wish I would have known, because I would have done things a little bit differently and not have to reinvent the wheel in some cases. So that, if you're looking for challenges to this translational research, I would say that in my personal experience, that's been one of the more significant challenges that I've encountered.
And I think you're absolutely right, Jim. The lack of transparency with the regulatory agencies, it's a black box, and you put something in and you never get those generative discussions. And part you can understand why, because they've got to be very, very cautious in terms of approving a drug. But I think there's a lot of scope for improvement there, yeah. So I want to move on to a slightly different topic. And Tony, could you tell us more about your work in comparative oncology and the use of biomarkers for clinical application of targeted oncology drugs?
Yeah, sure. Happy to. I think the interest in biomarkers started almost right away, because my residency project at Purdue was actually a biomarker study in the context of all that I described earlier. And just for way of background, biomarkers, a fairly general term, but applies to taking samples. They can be from, in the cancer case, the cancer itself, or circulating in the bloodstream or in the urine, or a surrogate biomarker like another piece of tissue that has an effect on the cancer itself, to give you information, which I tend to put into three broad categories: prognostically, so trying to figure out, in addition to some of the conventional ways we do it, what this cancer is going to do, how aggressively or not it's going to behave, independent of how we would approach it treatment-wise. Versus the second category I think of, which are predictive biomarkers. So I have a therapeutic and I know it's not going to work in absolutely every patient. So can I predict ahead of time which ones will respond and which ones will not, so I don't waste time for those cases that are not going to respond to the drug? And then there's a third category that I tend to put in the more optimization side of things, that's become really much more relevant in this newer era of targeted therapeutics. And that's sort of where my PhD took me from Purdue. So at Purdue, we were looking at COX-2 expression and prostaglandins in the tumor on and off inhibitor treatment, because only about 20% of dogs with bladder cancer would respond to that piroxicam drug. Could we find out if we could predict their ability to respond with COX-2 application in the tumor itself? When I went to Sunnybrook, this was in the era where the newer targeted drugs were just starting to get approved. So Gleevec for myeloid leukemia, Herceptin for breast cancer. And there was that recognition that, unlike the dogma for chemotherapy that most people are familiar with, where you give as much drug as you can without killing the patient, the maximum tolerated dose, that wasn't necessarily serving well as the way to apply a lot of these new targeted therapies. And so my work there was focused on some of these new kinase inhibitors and can we find the optimal biologic dose? And it was sort of focused on a target pathway called the epidermal growth factor receptor, or EGFR, which a lot of carcinomas, so a lot of human cancers, may have expression of, and it had a whole string of therapeutics associated with it. And we were able to show that you could titrate the response, particularly to the antibodies being made as treatments to individuals based on their expression of this receptor. And so the work was done in cell lines that had various expression levels, and then preclinical xenograft models of breast cancer, colorectal cancer. And then ultimately, we were able to modify a clinical trial that happened to be going on at the time in Toronto. And again, by way of background, the field was somewhat influenced by the fact that the first two drugs were really home runs. Like Herceptin worked if you had the target HER2 and it was very predictive, but that didn't really work out as well for EGFR. And there was a whole, the response rate in the first round of trials was modest at best, and they were second guessing whether EGFR expression was actually important or not. So we were able to get a subset of patients who did not have EGFR expression on their cancer, but had it in the body, and then take blood samples to look for these same biomarkers that we had been looking at in the lab. And so that sort of thesis ran the gamut from just looking at cell lines all the way into the human drug application. And it was interesting being a part of the Genentech and Pfizer discussions because, as was mentioned before, the stakes are really high going to these clinical trials, and it's not so as much, how much do we know? We don't know what we don't know about the target expression predicting response. They only enrolled a certain target population because you assume that that was the target that was necessary. And as we get so much more information about the makeup of tumors, genomically and otherwise, how do you design these investigations in order, do you take all comers and interrogate everybody, which is a larger trial, takes a lot more time and a lot more, or do you perhaps naively focus on the therapeutic marker you think is going to be most important? And so that biomarker research was really trying to answer, how do you optimize application of these drugs? And I'll sort of end on the fact that at that time, the first kinase inhibitors were being developed for veterinary use. So that was at the time where Palladia, that Chris mentioned earlier, was in the drug development pipeline, and we were working on the human correlate sunitinib and finding out that, how would we best apply that drug? And as we know, as Chris knows, the dose that we started with at Palladia wasn't the actual optimal dose that we ended up using. And in the drug development process, Cheryl London and others were able to use surrogate biomarkers to show that you can optimize the dose based on inhibiting the target with that drug. And that was something that the field hadn't really done a whole lot of at that time. They were just getting into the target modulation space. It had been so driven by phase 1, phase 2, phase 3, define your dose by optimal tolerability and toxicity, and trying to then evolve into target modulation. And so we were really kind of at the forefront of that, which is really quite interesting.
Great. Thank you, Tony. Christopher, can you tell us about your work on the immune landscape of cancer in dogs, and what translational methodologies did you apply in your work, and how will it benefit people and animals?
Yeah, so my DVSC project was really focused on the programmed death receptor and its ligand, or PD-1 and PD-L1, which, for those who don't know, has really gained a lot of traction in the human space, and certainly now in the veterinary space, as well as new potential antibodies are going to be coming to market soon. But the whole idea and kind of rationale for this is that we know that a lot of tumors kind of sit with a lot of immune cells, and for some reason, those immune cells don't kill the cancer cells. And the thought was, can we reprogram or provide a therapy that would potentially allow the immune system to go back and actually attack the cancer? And so by applying in humans these anti-PD-1 or PD-L1 therapies, we effectively take the brakes off the immune system and allow it to attack the cancer. that was, really shown in human lung cancer. My own grandfather was on that type of therapy and did fantastically well. But we see it now very commonly in breast cancers and so on. And so our goal was to look at that in canine urothelial carcinoma with the thoughts that not only had there been lots of data in humans to show how immunogenic the tumor was and that they expressed these receptors and ligands, but no real work had been done yet in dogs. And so we were able to look at not only cell lines, but clinical patients for their expression of these things. It wasn't like anything revolutionary, but what it did do was kind of lead to the thought that it's certainly an immunogenic tumor as we expected in canines, and again would be really a translational model. And now with the work at Sunnybrook, I think similar to Tony, whatever you do in your residency, you kind of feed into probably what you're going to work on moving forward. And certainly for me, looking at the immune environment, tumor infiltrating lymphocytes, which is a huge thing in human breast cancer and is becoming definitely more exciting in the veterinary space as prognostic markers or markers for treatment response and biomarkers, as Tony alluded to, I think is going to be really huge. And so what we're hoping to do is, obviously we're looking at humans with breast cancer already and looking at whether TILs can actually predict kind of the response to neoadjuvant chemotherapy in women with breast cancer. But then could we start to apply that to dogs? We know that immune response may play a role with radiation therapy. So our thoughts are maybe we could start looking at patients, canine patients with oral melanoma, and does TIL infiltration play a role in radiation response or overall survival? So again, really all this kind of biomarker data. And the other thing that the group's doing that I work with is that we're using artificial intelligence to then take that to the even further level, not just looking at the number of TILs that are kind of in this tumor microenvironment, but even directionality. What direction are those TILs facing, and how are they interacting with the cancer cell, and does that provide any sort of impact? And then what TIL is it, and to what degree? So lots of cool things, lots of ways that we can start to use dogs as a surrogate model and say, if, as Jim alluded to, if we can take a whole bunch of cats with triple negative breast cancer and take their biopsy samples and look at that as a prognostic biomarker, model that in dogs or cats, and then apply that back to humans, then you've got a lot of translational relevance there.
Great. Thank you, Christopher. Steffi, could you tell us about your thesis on lung cancer in humans and canines, and how you visualize primary and metastatic tumors in animals during cancer surgery, and how that's going to translate to humans?
So we've been working on image-guided surgery in humans and canines for a while, and also with lung cancer. So what we do is we use non-specific imaging agents, such as things called ICG, which is or indocyanine green, to visualize tumors and lymph nodes. So the imaging agent would be injected, and we would visualize in the tumor bed with a near-infrared light, and it would fluoresce. So due to the non-specificity of the dye, it is not commonly used in surgical practice. So what we're trying to do, the next step, is to improve surgical resections to develop a targeted version of indocyanine green, where it'll be conjugated to a ligand that can bind to an overexpressed receptor in lung cancer. And so that would only fluoresce any cancerous tissue, while the background would be not fluorescent. So the potential to use something like a very highly selective dye for image-guided surgery for lung cancer really allows us to improve the prognosis of the patient and any patient outcomes, because you can get better margin resections, delineate lesions in lung cancer and tumor nodules, and potentially even find metastatic lymph nodes. And so overall, that safety profile of the dye, because it is an FDA-approved dye, plus the comparative research component of using the dye for canine patients and then also human patients, really allows us to use the imaging probe and push it further along in the pipeline to get it to human clinical trials. So that's kind of the goal of the project.
Great. Thank you, Steffi. Jim, can you describe how you use animal models to study reproductive function and to model late-stage ovarian cancer, and what the impact of your research is going to have on humans?
Yeah, absolutely, Howard. So, we're interested, as I mentioned before, about modeling late-stage disease, and our most models of disease that we've generated are orthotopic syngeneic models, where we put the tumor cells in the appropriate tumor microenvironments in an immunocompetent model. And I think that those are really two really critical factors, is that we found that putting the cells in the ovarian microenvironment and allowing them to progress in the tissue microenvironment that they normally would has had significant impact and benefit on the model. And then also having an intact immune system has been utterly critical for normal tumor development. So, our approach is solid tumors, in order to grow, need to develop an adequate blood supply, and they do that by aggressively recruiting peripheral blood vessels to sprout and migrate and penetrate the tumor and develop its own vascular supply. But the problem is that angiogenic program is so aggressive that the tumor blood vessels are actually very dysfunctional and amorphically formed. So the tumor actually has a very poor blood supply. So our approach is to just kind of specifically target those dysfunctional tumor blood vessels, prune them back, so destroy the dysfunctional tumor vessels, prune back to healthy parental blood supply. So we end up with a small tumor that has a fantastic vascular perfusion. And we've been able to exploit that enhanced vascular supply by enhancing the uptake and efficacy of other therapies that I mentioned before. Chemotherapies, we've done some work with Tony on that. Immunotherapies, one of the problems with immunotherapies is that you stimulate the intratumoral immune system and you recruit these activated immune cells, but then they can't get into the tumor because the highway getting into the tumors is corrupt. So we've been able to really to show that once we activate the intratumoral immune system, that we get about a 500% increase in the uptake of immune cells into the tumor core. More importantly, that we found is that when we normalize the tumor blood vessels, we get rid of the leakiness and the leakage of fluid inside the tumor, which creates a very highly pressurized structure. So the interstitial fluid pressure in a tumor is typically very high. So if you think of it from a therapeutic perspective, we deliver intravenous therapies, immunotherapies, chemotherapies, and relying on a completely dysfunctional supply system, so that the perfusion of the tumor is challenged. But then we're also trying to get drugs passively into a very highly pressurized structure, and so it's counterproductive, very counterproductive. But the other thing we find is that with the high pressure inside the tumor lymphatics, which drain the fluid from the tumor, are compressed and collapsed. So it's a feed-forward mechanism. So that contributes to the continued elevated pressure within the tumor. But from a therapeutic perspective, it's those tumor lymphatics that allow migration of activated immune cells and checkpoint molecules to drain from the tumor into the draining lymph node, and that's what educates the peripheral immune system. So if you have a solid tumor that has collapsed lymphatics, you're not getting movement of those immune cells to the draining lymph node, and you have a very muted peripheral immune response. And that's why there's theories out there that's why the success of immunotherapies in general has been less than originally anticipated. So with our therapeutic approach, we found that we dramatically increase the number of immune cells that drain to the draining lymph node, and we get checkpoint inhibitors, so anti-CTLA4 that we mark with fluorochromes. We now see the draining lymph nodes really kind of beautifully decorated with all these immune cells and checkpoint inhibitors, and we see a much more robust immune response to the tumors, and we've been able to induce, at least in our preclinical models, this curative approach where we have complete eradication of metastatic disease in our models of advanced stage ovarian and pancreatic cancer. So we're excited about that, and as I said before, we're translating this to the companion animal clinical space. We want to look at these exact same outcomes in companions, and we've done a healthy dog study and worked with Tony and Michelle Oblak, and Charlie McKenna was fantastic and helped facilitating that early initial safety study. Now we want to move on and look at traditional chemotherapies, but also as Chris is talking about, immunotherapies, checkpoint inhibitors, and really providing the opportunity for these therapies to have the highest potential for therapeutic success.
Great. Thank you, Jim. We're beginning to get a little bit short on time, so I'm going to skip forward in our, I guess the panel members know we have a script that we're following, and I'm going to skip forward to the last section on advice for graduate students, because I think that's important for the audience to hear your impression on how to get where you want to be, and then hopefully we'll still have a little bit of time left for questions. So, Jim, can you comment on why OVC is uniquely positioned for translational research and how graduate students can get involved in translational research?
Yeah, and I'll try and keep this short, Howard. OVC is the only place this is going to happen. The OVC has these really amazing models with the companion animal cancer patients and patients with other diseases, cardiovascular disease, neuropathies, other things, not just cancer. I keep talking about cancer, but it's applicable to other disease states. But more importantly, we have amazing people here. We have our clinical oncologists, surgical oncologists. We have people here that are amazing at running companion animal clinical trials, and I think the opportunities that are here have been so underappreciated and underutilized. So I think our translational opportunities here we're just at the precipice of really taking off with enhancing and maximizing our translational impact. And the human clinicians that we work with that are doing human cancer trials are starting to notice the opportunities. We've done a lot of work, and there's an extreme amount of interest there. So, with respect to what the grad students can do, I think that, if the grad students are working in an area that has a translational pipeline or trajectory, is to, try and talk to your supervisor and talk about how you can get involved in some of this translational research that has a clinical outcome. Is the disease you're working on, or the mechanism you're working on, does it have a veterinary application? Is there an opportunity to work with some of the clinical veterinary people here and as a way to learn more about the biology of the disease, and then ultimately translate that potentially, if that's an interest, to the human space. So I think there's amazing opportunities here with our veterinary clinicians that provides amazing, unique, and really impactful opportunities.
Great, thank you, Jim. Steffi, could you tell us a bit about your experience as a graduate student spanning 2 different departments at OVC, and what did you expect going in, and what surprised you? I assume something surprised you.
So when I began my program, it was a little daunting since I did have a very biochemistry background rather than any clinical research experience. But then having 2 co-advisor, like one from biomedical science and clinical studies, it really helped me have like a balanced understanding of benchtop research and then clinical research. So it was very interesting and surprising to always have to think of the clinical significance when planning projects or experiments, and everything had like an end goal at a clinical level, because then it would be meaningless on that side of things. And then it was nice to see things, you would see things at a macroscopic level, and then a very more scaled down level in basic science. So having both of those, it's a very good perspective to have. And I think grad students, if you're wanting to go into kind of translational research, it's, have an open mind and kind of be ready to answer questions in a certain way that has a bigger outcome and a bigger picture rather than small details.
Great, thank you, Steffi. Tony, you're cross-appointed between 2 departments in OVC. How's that benefited your research?
Oh, great question. For me, it was incredibly valuable when I first got here to be based out of, clinical studies is my home department, and biomed is where my office and lab are in the cross appointment. And there's such a strong cluster of cancer researchers in biomed that I knew I would be placed in amongst it to be able to have conversations as an early faculty member, develop a research program. And, having said that, I think OVC as a collaborative environment is very strong and stronger than other places I've been. And this was a bit of a safety net that's worked out really well for me to be able to integrate into a cancer research group, both at the departmental level and within the lab, because it's a multi-investigator lab all together on the 3rd floor. And, I collaborate heavily with people in pathobiology. And like I said, it's been a really great experience to have the investigators in biomed as well as in clinical studies. And to follow on the last points, it's for my students as well, although I might not have thought that going in, because I have students who are in clinical studies, primarily DVSCs, some master students, and then biomedical graduate students and PhDs and master students, and they work together in the research space, as was said. So I think the clinical trainees would agree that having that access to the biomedical grad students when they have their protected research time has been really valuable. And certainly the biomed research students, I think, really appreciate interacting with the clinical grad students and the clinical environment. So there is that cross influence between the clinical focused graduate students and the biomedical focus, knowing that there's that bigger picture that was mentioned before.
Great, thank you, Tony. Christopher, can you tell us more about your role at the Toronto Veterinary Emergency and Referral Hospital and the Sunnybrook Research Institute, and what path did you take in transitioning from being an OVC graduate to getting into these positions, and what advice would you have for current graduate students as they embark on their careers? And I think this is really critical because not all graduate students are going to end up in academia, which is the classical path, and you've gone on a different path. So I think your response is going to be enlightening for many in the audience.
Thanks, Howard. Yeah, so I mean, as everyone's probably well aware, like I do wear two hats, right? And the first is that the majority of my time is really to treating clinical patients with cancer here in Toronto, where I spend most of my week seeing dogs and cats with cancer. The other part of my time is really as much time as I can devote to it is as a postdoctoral fellow at the Sunnybrook Research Institute, and that role really came about out of a relationship that I formed during my residency, quite casually, in fact. And after talking for some period of time, my current mentor there, Dr. Tran, I still remember the distinct day where he was like, when we first started talking, I didn't even realize dogs got cancer, but like, how silly was that? And it's really kind of that little switch point has now led to us talking not only collectively within the Tran lab, but then also with other folks at the SRI, hoping that we can start to build this more, and hopefully in for future collaborations with OVC and others, that we can start to really bring this into a real model that can be used more routinely. So my path really, I guess, was a little unconventional in the sense that I think it highlights one of my pieces of advice to grad students, and that is, really don't be afraid to make relationships, and don't be afraid to email someone out of interest and ask a lot of questions. That you never know what one email, one relationship, one casual meet and greet is going to set up for you in the future. I think if you can show that you're passionate about something and a mentor can see that in you, I think they're always willing to take you on. And I think we've also got, and I think Jim, Tony, and Steffi really kind of all highlighted this, that we all have so much to learn from one another. And being in Tony's lab, I think really highlighted that for me as well as a DBSC student, that I learned so much from the folks that were more focused on basic science and not clinically focused, and I hope they felt the same way. And that mutual relationship has really kind of set me forward on my path in the future. So expose yourself to everything, get excited. And I think in a lot of my last piece of advice would just be that we do always try to, I think, to strive for perfection, both in a clinical and research setting. But I think it's really the moments where we're most vulnerable, where we're incorrect, and where we show some humility, that we actually can really, really divulge and learn from one another, and learn really quickly and do some really amazing things.
Great. Thank you, Christopher. Formally, we're at the point in the this morning's discussion that we're going to open it up for questions from the floor. I don't see any questions in the chat channel as yet. So if anybody in the audience has a question, and I see one hand up, Janet Bila-Mafazi, would you like to turn on your microphone and ask your question?
Yes, please. Thank you to everybody on the panel. It's been a very interesting morning, and I have 11 billion questions, but I will restrict myself to, well, I have one for Chris and I have one for Jim. So I'll ask Jim his first, because it's, as all my questions are, super selfish. And I'm asking from the basis of someone who can't yet afford to do an explant study, or where, since I study asthma in horses and the mechanisms behind lung inflammation in general through macrophages, I'm just wondering, because you study things in the environment that they're meant to be in, what are your thoughts on the role or utility of those models that investigate one element at a time? Yeah.
Yeah, no, yeah, that's a good question, right? It's you know, how tightly do you distill down your research question? But I still, to be honest, and I still think that the context, the environmental context is utterly critical and relevant, regardless of how small the question is you're asking. You know, in the interest of time, I won't go into the specific biology of the example, but by putting cells in the right environment, we actually learned a tremendous amount. Beyond the traditional models before that were inject cells in the abdomen, they form lots of metastatic balls of cells, and they considered that metastatic cancer. And we found that those were just small spheroids of cells. They weren't actually heterogeneously well differentiated cells. So I think it just highlights out the importance that no matter how mechanistic your question is, the appropriate environment is still is critically important, in my opinion.
Great. And Janet, you had a second question?
I will work towards tissue explant models very shortly. Thank you.
Janet, you had a second question for Christopher, I think?
I do, and it has to do with the TILs, Chris. If we're using cells that are switched on and meant to kill stuff, how do we stop that super delta force from going haywire, or when they've done their job, what happens?
Yeah, it's a really great question, and to be honest, I think the real answer is we don't know, and that kind of falls in line, clinically speaking, that, you know, I was just at the European Society of Veterinary Oncology conference, ESVONC, in Italy, and some of the human researchers were talking about immunotherapies in their space, and one of my questions for them, and I think there was a talk recently through Guelph that I joined as well, that we're trying to figure out, and we still don't know, and we, I mean the royal we, both veterinary and human oncologists, about how long do we treat patients for with immunotherapy, or what is the correct dosing regimen, and is there a time that we should stop? Do we continue immunotherapeutics continually because we think that, you know, otherwise we're going to confer some form of resistance in the traditional sense that we would with other types of chemo? I think the real answer is we don't know. And I'm sorry, I hope that answers one part of your question. But then your other about, you know, how do you kind of, once you've turned on the immune system, can you turn it off? And I guess effectively by stopping immunotherapy, you may do so. But one of the other clinical concerns is that you can see patients that either get overt, you know, really bad sort of anaphylactic-like reactions to these immunotherapies, or we can get something called hyperprogression, which is essentially where, for whatever reason, whether turning on that immune system or whether the therapy didn't work, we get these tumors that progress extremely rapidly and may progress more quickly than if we hadn't given that patient immunotherapy. And all the mechanisms behind that are still being studied, aren't really completely understood, but I think knowing and looking at TILs, as you mentioned, I think will be a very interesting biomarker as time goes on.
Thank you.
Great. Thank you, Christopher, and thank you, Janet, for your question. We're pretty much out of time now. So what I'd like to do now is firstly, you know, thank the panel for your incredible contributions this morning. This has been a fantastic discussion. And as I think, you know, certainly some of the panel know, I've been working with OVC in the background in terms of, you know, growing our research application, particularly in translational medicine. And I've been writing a list of, you know, new areas where I think we can talk about other potential opportunities to broaden that collaboration with industry. So for me, this has been a very valuable discussion. And I think, you know, we've mostly talked about cancer this morning, but I think virtually all of the comments that have been made could very easily translate, and I think translates the appropriate word, to other disease areas. So I hope for the audience that you found this helpful, and I'm going to stick my neck out a little bit here and say that all of the panel would be happy to engage in later discussion. We're all within the University of Guelph. We all know how to find out someone's email, and please reach out and ask those follow-up questions, because it's important that we work as a community and grow together in terms of advancing our research and advancing our careers for the benefit of both animals and humans. So with that final comment, I'd like to thank everyone again for attending this morning. As I say, this has been extremely rewarding for me, and I hope it's been rewarding for the audience. And I know we've got more going on in the general program this morning, so I want to give everybody time to probably have a bio break or something before the next portion of the morning starts in 10 minutes time. So thank you very much, everybody, and enjoy the rest of your day.